Good morning, everyone. This is Patricia Townsend from Washington State University, welcoming you to our fifth in our Hardwood Biofuels webinar series. If you are just joining us, could you please answer our poll questions on what category describes where you are coming from today, and what is your familiarity with using endophytes in hybrid poplars? We will be starting with our presenters momentarily. To get started, I'm going to tell you a little bit about our project, Advanced Hardwood Biofuels Northwest. We're taking purpose-grown hybrid poplar trees and we're turning them into drop-in replacement transportation fuels and bio-based chemicals. Advanced Hardwood Biofuels Northwest is a consortium of university and industry partners, and is divided into 5 different teams: feedstock conversion, sustainability, education, and extension. Today we have some people from our feedstock team that will be speaking to us. We have Sharon Doty, who is a professor at University of Washington, and she's going to be talking to us about increasing growth with fewer inputs. And then we also have Posey Busby, who is a postdoctoral fellow at the University of Washington and University of Idaho, and she will be speaking to us about increasing disease resistance. And the overall title of their webinar is Endophytes to Increase Sustainability and Disease Resistance.
Hey, this is Sharon Doty. So you may have heard of the human microbiome that studies the microorganisms that live within human beings. We'll be talking about the plant microbiome. So there are many different microbial communities that reside within a plant, and this includes the fungi, bacteria, and archaea. And they're throughout the plant. They're within the leaves and the stems and the roots of the plant. So these are defined as not being pathogenic. These are microorganisms. Oh. Sorry, technical difficulties. These are defined as microorganisms that do not cause disease. And yeah. These are mutualists. So they're symbiotic partners that benefit the plant in multiple ways. They can provide nutrients to the plant through nitrogen fixation and solubilization of phosphate. They can increase stress tolerance, helping the plant to be tolerant to heat and salt and drought. Also pathogen resistance that Dr. Busby is going to be talking about. Also pollutant degradation. So if the plant takes up pollutants that are toxic to the plant, then some of these microorganisms can degrade those pollutants. And they also help with plant growth promotion overall. So some of them produce phytohormones. So the way that we isolate endophytes for study is you can take any of the plant tissues, the plant organs, the leaves, stems, and roots, and surface sterilize them with something like a dilute bleach solution or iodine or mercuric chloride to clean the surface, so that we're looking at the microorganisms that reside within those organs. And then if you just simply plate that out, then we get a variety of microorganisms that grow out from within the plant tissue. So we can study them in isolated culture. So one of the reasons why I study endophytes is to try to have an alternative, biologically or an environmentally sound alternative to using chemical fertilizers. So plants require nitrogen, and pointer here. So this is showing plants grown without fertilizer, and then this is an increasing amount of fertilizer. So plants grow better, and they're greener and healthier if they have nitrogen fertilizer. The problem is that synthesized nitrogen fertilizer requires high levels of fossil fuels. And as these become limiting, then the cost of fertilizer also rises. In addition to these problems, there are multiple environmental costs to using chemical fertilizers. One is that the excess nitrogen fertilizer and also phosphate enters our waterways and can lead to disruption of those ecosystems, algal blooms and things like that. Another serious problem is that the excess fertilizer in the soils gets converted by the soil bacteria to nitrous oxide, which is a very potent greenhouse gas. So there are multiple reasons why we'd like to get away from our dependence on chemical fertilizers. So a natural alternative to chemical fertilizers is dependent on biological nitrogen fixation. And this is where there are certain microorganisms that can convert the nitrogen gas that's in our air. Okay, so our atmosphere is about 80% dinitrogen gas, but this is an inert molecule, and only certain microorganisms have this enzyme called nitrogenase that can reduce this dinitrogen gas into ammonia, which is the usable form. So this is what is in fertilizer, ammonia nitrate. This is a very energetically expensive reaction requiring high levels of ATP, the energy currency of a cell. And so it's often plant associated so that the plant can be providing the microorganism with sugars from photosynthesis, and then the microorganism can provide the plant with the fixed nitrogen. The most well understood and well characterized of these nitrogen fixation symbiosis with plants is through these root nodules. So this is rhizobium in legumes, and also frankia in alder trees and similar plants. And so it's well known that these types of plants can benefit from this symbiosis and get enough of their nitrogen. However, this is a very limited group of plants. So only the legumes, such as peas and beans and lupin, and then alder trees, have these root nodule symbioses. So just recently, in the about 1990s, it became clear that there's another way that plants can obtain this naturally fixed nitrogen, and this is through nitrogen fixing or diazotrophic endophytes. So this is nitrogen fixation without root nodules. So there's no structure within the plants that we can see. It's the microorganisms are within the plants. They can be in the roots, the stems, and the leaves. It was shown first very clearly in sugarcane as well as callar grass. So when plants are grown in natural conditions, some of them develop this symbiosis with these endophytes, and it can provide a substantial amount of the necessary nitrogen for the plant. Another case that was shown was in African sweet potatoes. And then what I've been studying are the poplar and willow in their natural conditions. So the natural environment for poplar and willow is often in these riparian zones. And some of these can be dominated by this cobble, which it's just rocks. There's no soil, there's no organic material there at all. And these are natural river systems, so they're not polluted with chemical fertilizers. So that these plants are completely dependent on symbiosis in order to get their necessary nutrients. So I've isolated endophytes from poplar and willow, and we found a nice variety of different microbial species. We've shown that many of these isolates are able to fix nitrogen from the air. So we've shown this through the ability of these microorganisms to grow in nitrogen-limited medium. They have the nitrogenase gene that encodes that nitrogenase enzyme for reduction of the gas. We've also shown through isotopic analysis that they are fixing nitrogen. I don't know if you can see this at all, but another attribute that these microorganisms have is the ability to solubilize phosphate. There are two major macronutrients that plants require. So one is nitrogen and the other is phosphate. In this medium, the phosphate is insoluble. It's calcium phosphate. But if the microorganism is able to solubilize the phosphate, then we get this clear zone. So we've shown that many of these endophytes, so in this graph that you can't see very clearly, they have a very strong ability to solubilize that phosphate. The controls were down here that are not able to solubilize the phosphate. Here's a picture of one of the controls. So if it can't solubilize the phosphate, the microorganism can't really grow well on this medium either. And another benefit that the endophytes have is many of them are able to produce phytohormones. So one of the common ones that they produce is auxin. This is indole acetic acid. Auxin is a rooting hormone. So you can see from this graph that many of the endophytes produce fairly high levels of this necessary hormone. So we've shown then in culture that the endophytes from the wild poplar and also from willow can fix the atmospheric nitrogen into ammonia. They can solubilize phosphate, and they produce phytohormones. So then the key question here is, can we add these back to cultivated poplar for increasing the growth and health with fewer inputs? So in order to determine if we have successfully recolonized this poplar, we use fluorescent tags. So we can add green fluorescent protein or red fluorescent protein or other colors to the different endophytes, and then inoculate or add these endophytes back into poplar, and then we can track their ability to recolonize. So in these graphs, hopefully you can see more clearly than we have here. This is a longitudinal section of the poplar. So this is a poplar cell here and a poplar cell here. And you can see that the bacteria are all within these intercellular spaces. So we know that the bacteria here is what we added because of the green and the red fluorescence in this picture here. In some cases, the bacteria are all grouped together in the intercellular spaces. In other cases, they're in the vascular tissue, so in the veins of the plant, so the xylem and phloem. This work was all done by Amy Baum. She's a PhD graduate student in my lab who's part of this AFRI project. So we've shown that we are able to recolonize poplar, so hybrid poplar, with the endophytes from wild poplar. And so here we're showing that it does have these pronounced benefits to the plant when we do this. So this is a poplar clone that's very recalcitrant to rooting. So it was a hardwood cutting. When we add the microorganisms from wild poplar, then we see pronounced increased rooting. In this picture here, it's showing the in hydroponics it also works. So this is without the added microbes, and this is with the microbes. These were added to the hardwood cuttings, and then they were placed in this hydroponic medium. So it shows that probably because of that phytohormone production by the endophytes that the plants are now able to have the increased rooting. In this graph, we're showing this is Populus trichocarpa, Nisqually 1. This is the clone of poplar that had its genome sequenced. When we do the same sort of experiment here, we're seeing almost a doubling of the root mass of the plant in this greenhouse study. So this is the uninoculated control and average of the cuttings that were not exposed to the wild endophytes, but they do still have the endophytes um, that were in the clone itself. So it's adding the wild endophytes that have this pronounced effect on growth. And these effects are long-lasting. So when we inoculate the hardwood cuttings with the wild endophytes, and then we take cuttings of these shoots and put them in the hydroponic medium, and this is when we're seeing this effect. So even though we added the microorganism at this stage, its cuttings, even up to a full year later, is what we've tested so far, we're still seeing this pronounced growth effect. So it's a long-lasting positive effect. So in terms of nitrogen fixation, showing if these endophytes from wild poplar are able to provide this necessary nutrient to the plants, we can see in wild poplar, of course, that it's able to grow without any fertilizer and without any soil, any organic material. So they're thriving on nothing but rocks. When we take cuttings of poplar and grow them in hydroponic medium without any ammonia or nitrate, you can see that the plants are still green and healthy. When we do the same experiment with a cutting of tobacco plant, however, the tobacco is severely stunted and chlorotic, yellow, it's not growing, compared to a cutting of tobacco in normal hydroponic medium with ammonium and nitrate. So clearly the poplar is able to get its nitrogen from a source other than the hydroponic solution. It's also been shown by various different growers of poplar in plantations scale that they often don't even need to add fertilizer. So they're showing that in these cases there are endophytes already in their poplar that enable them to grow without fertilizer. We've done greenhouse assays showing that nitrogen fixation is occurring in poplar. In this particular case, it was a 15N dilution assay where the plants are first labeled with this 15N. And then if you see a decrease, this is comparing uninoculated with inoculated with the wild endophytes. When you see a decrease in the 15N label, then that means it was taking nitrogen from the air. The normal isotope of nitrogen gas from the air is 14N, so it's being diluted through nitrogen fixation. So we've shown that about 65% of the foliar, so the leaf nitrogen, was through biological nitrogen fixation. We've also recently shown through direct 15N incorporation, so where we have cuttings from wild poplar in a sealed vessel dosed with 15N gas, that we are getting 15N incorporation. So we've shown in multiple ways now that poplar is able to get its nitrogen through nitrogen fixation. This results in increased greenness of the plants. So this is poplar grown without any ammonia or nitrate. So this is a measure of the greenness, the measure of the chlorophyll content of the plants. When we add different mixtures of the poplar endophytes from the wild or willow, we see a significant increase in the amount of chlorophyll that the plants have. And another benefit of these endophytes from wild poplar is that it increases the drought tolerance of the plant. In this picture, this is with grasses. So grasses are also benefiting from the poplar endophytes. If we add the endophytes to grasses, then they're able to tolerate drought significantly better than grasses that don't have the poplar endophytes. A similar experiment was done with poplar cuttings. So this is hybrid poplar from Greenwood Resources. And when we add this the endophytes from wild poplar, then the plants are able to tolerate and even thrive with less water than the uninoculated controls. So plants you won't find plants that don't have endophytes in them. But we think that these endophytes from these environments, from wild poplar, are special, and that they promote the growth and fix nitrogen better than the average endophytes, we'll say, because we think it's because there's much stronger selection for this mutualism in this type of environment. In this environment where the plants are growing in this cobble without any organic matter, there's no soil, the plants are completely dependent on their symbiotic partners to get the nutrients that they need. So there are multiple implications for this research. One is with the bioenergy, so production of sufficient biomass to make biofuels. So this is an important part of our AFRI Advanced Hardwood Biomass Grant, is to develop ways so that we can grow poplar in very large scale without any environmental damage. So by using natural symbiotic partners from wild poplar, we can reduce the amount of chemical fertilizer that's required and even the amount of water that's required. Because these endophytes from wild poplar also work in agricultural crops, including corn, rice, grasses, tomatoes, bell peppers, there's potential that we could reduce the amount of chemical fertilizer that's applied to fields and then help clean our river systems and prevent these dead zones that result from excess fertilizer entering our river systems. And also it can impact climate change. So plants are able to sequester or do more photosynthesis, more removal of carbon dioxide from the atmosphere if nutrients such as nitrogen are not limiting. And so if we have these symbiotic partners, we may be able to increase photosynthesis. And using more chemical fertilizers in order to produce more biomass for bioenergy is inconsistent with climate change mitigation because of this issue of the chemical fertilizers producing nitrous oxide, the potent greenhouse gas. So it makes much more sense to use natural symbiosis for nitrogen fixation rather than chemical fertilizers. Okay, and so we've shown that these endophytes can promote plant growth naturally through this improved nutrient acquisition, through nitrogen fixation, phosphate solubilization, and also phytohormone production. So this work is done, the work that I presented today was mostly done by Dr. Zareen Khan, a research scientist in my lab, Amy Baum, the PhD graduate student on this AFRI project, and the undergraduate student researchers on this project were Andrew, Neil, Alex, Megan, and Beverly, and the funding was from our AFRI project. So I'm going to turn this over now to Dr. Posey Busby, and then I'd be happy to answer your questions at the end of her talk. Thank you.
Okay, so I'm going to start right in on part 2 of the webinar, endophytes to increase disease resistance. My name again is Posey Busby. I'm a postdoctoral fellow here at the University of Washington and at the University of Idaho. I'm going to start by introducing the research team that I work with on this project. In addition to myself, Josh Miller is a graduate student in the lab of George Newcomb at the University of Idaho. He started on the project a few years ago when I also started. He was an undergraduate at the time and has now transitioned successfully to the master's program. Shannon Fraser is a technician working on the project, and the three of us work under the mentorship of George Newcomb, professor at University of Idaho. And in addition to working with the Advanced Hardwood Biofuels group, we also have a number of other collaborators and additional funding sources that are listed here. Okay, so I want to start where Sharon left off in the populous microbiome. And like Sharon, I'm very interested in plant microbes that are beneficial both to plant growth and to plant immunity, and that thus could potentially be used to increase sustainability in feedstock production for biofuels. And most of Sharon's work that she talked about focuses on bacteria, but the work that I'm going to talk about now is focused more on fungi within the plant microbiome. More specifically, I study fungal leaf endophytes. So these are fungi that live within the above ground portions of the plant, of the poplar, specifically. These fungi live within plant leaves, and as Sharon described, they are defined really by the fact that they're non-pathogenic, so they don't cause disease in the leaves where they live. Unlike some of the systemic bacterial endophytes that Sharon talked about that are found throughout the plant tissues and then that can be transferred from one cutting to the next, fungal leaf endophytes form localized infections within their leaves. So one leaf may have a very different community than a leaf on the same tree even. We also know that fungal leaf endophytes are ubiquitous. So far they've been found in all land plants that have been surveyed, and they're hyperdiverse. So within an individual leaf, it's not as if you might find 1 or 2 species of fungi, you might find 100. So really incredible diversity of these fungal leaf endophytes. And they live side by side, a very different group of fungi in poplar leaves, and that group of fungi are pathogens. Now, of course, pathogens are, these are organisms that you're probably more familiar with. Pathogens cause disease in their host plants. And by causing disease, they reduce plant growth, and in a plantation setting, then, they can reduce overall yield. So it's for these reasons that disease is really the most serious biotic threat to the economic production of populous feedstock for bioenergy. Now, in the Pacific Northwest, there are a suite of different pathogens that affect poplar both in the wild and in plantations. Those include a couple species of Melampsora rust, Melampsora occidentalis, and the hybrid Melampsora, Melampsora columbiana. And also some additional necrotrophic pathogens like Septoria populicola and Marsanina populi. Now, given that these pathogens are living side by side very diverse endophyte communities, they must interact on some level. And so this raises the question of whether some endophytes in these communities may have antagonistic interactions with those pathogens. And if so, then that raises a second question, which is, can we use those particular endophytes to increase disease resistance, in poplar plantations? So quickly, I can give an example, and that is that there are fungal leaf endophytes in poplar plants that are antagonistic specifically by mycoparasitizing rust. And so if we can harness these mycoparasites of pathogens, we can potentially increase disease resistance in plantations in ways that, that complement traditional methods of breeding for genetic resistance. So the first study that's really addressed this question of the potential of endophytes to be used as defense inoculants in plantations is a study by Anil Raghavendra and George Newcomb, published last year in the New Phytologist. And specifically, they looked at different fungal leaf endophytes of populace and evaluated their effects on rust. And so what they did was they took 4 different endophytes that were collected from a tree in the wild, and they tested them against rust by first inoculating plants with individual endophytes, and then subsequently inoculating those same plants with Melampsora rust. And they compared rust that developed in those endophyte-treated plants to rust in control plants that didn't get an endophyte, but that just got the rust. And what they found was that all 4 endophytes that they tested, shown here on the x-axis, all 4 endophytes significantly reduced rust, which the rust severity is shown on the y-axis, and some up to 16 times. So these were very dramatic effects of fungal leaf endophytes enhancing resistance in their host plants. And so what we'd like to do now is look at these types of interactions between fungal leaf endophytes and pathogens at a much broader scale. So look beyond 4 individual endophytes that were collected from a single tree in the wild, and look at fungal leaf endophyte communities and pathogen communities throughout the Pacific Northwest and what their interactions are like with pathogens. So the current research aims are first to characterize fungal leaf endophytes and pathogens of Populus trichocarpa, both in the wild and in plantations in the Pacific Northwest, where the biofuel industry is growing. Two, to evaluate interactions among poplar trees, endophytes, and pathogens in those areas. And then finally, once we have a good sense of what the fungal communities look like in plantations and then in the wild, and what the interactions are, what type of interactions we observe, we can investigate the potential to harness particular endophytes to increase disease resistance in plantations. So let's start with the first study aim, characterizing fungal leaf endophytes and pathogens of Populus trichocarpa in the Pacific Northwest. So to do this, we've been working in the study area shown here. I think many of you will recognize this. This is mostly Washington State here, the core of the Pacific Northwest. Also southern Canada, parts of Idaho and Montana and Oregon. And in this area, we're working both in field trials like Pilchuck and Jefferson, but also in wild populations of Populus trichocarpa. And the reason for this is that these wild populations, like the Skagit River population here, these wild populations will really be the source of inoculum for plantations or for field trials, in the sense that both pathogens and beneficial endophytes will come from these wild populations. So we want to know what the source population looks like. A second reason to study wild populations is that, as you can see, the populations that we've selected span from west of the Cascades, where we find very wet conditions, to east of the Cascades, where conditions are much drier. And we know that this environmental gradient very strongly affects pathogens, particularly west of the Cascades. We have, in these very wet sites, we have high disease pressure. And east of the Cascades, where it's very dry, we have lower disease pressure. And so we want to know whether that type of environmental gradient also influences fungal leaf endophytes and their interactions with pathogens. Okay, so to begin to characterize those fungal leaf endophyte communities in the wild, what we do is we go out to those wild stands and we sample leaves. So this is an example of a poplar stand here. And then we bring those leaves back into the lab and we surface sterilize them, as Sharon described, in a bleach solution. And then we take those leaves and we grow the endophytes out of them. So essentially, we put them in culture, and we wait for endophytes to grow out of the leaves, and then we can isolate individual endophytes and grow them in pure cultures on medium shown here. And then we can look at them microscopically to determine the identity of those individual endophytes. So these pictures here show a new species of penicillium that we isolated from poplar leaves. And you can see here, these are the spore-producing structures, the conidiophores of the penicillium. And then, these are the spores of, here's the conidiophore here. And then here are the spores, the asexual spores of penicillium. So by identifying species both microscopically and using DNA sequences, we can characterize these fungal leaf endophyte communities across the different study area. And we find that there are community differences east and west of the Cascades. So this figure here, what I'm showing you is an ordination analysis of endophyte communities of Populus trichocarpa, where each dot represents a different tree population or study site in the wild. And so these black dots are western river valleys here. The open circles are eastern river valleys. And so what you can see is in this ordination space, where the endophyte communities are summarized by simply 2 dimensions, we see that there's spatial separation. And points closer together have endophyte communities that are more similar, and points farther apart have endophyte communities that are more dissimilar. So we see east and west, we see there are, in fact, differences in the endophyte communities. We see more similarity among western sites than among eastern sites. And these lines here, these are vector analyses that show the different species in those communities that seem to be driving the communities apart. And in addition to different species, in addition to endophyte species differing east and west of the Cascades and creating these community differences, we also have environmental gradients that are important for structuring differences in endophyte communities. Specifically, rainfall is highly significant in terms of driving these community differences. So given that we see different endophytes in different places, the next question becomes whether we actually see different types of interactions between endophytes and pathogens in those different places. So the second study aim is to evaluate these interactions among trees, endophytes, and pathogens across the study area. And to do this, we're conducting a series of double inoculation experiments. And what this involves, again, is bringing isolates in from the wild, from all of these 12 different study sites, culturing the leaves, growing out pure isolates of different endophytes, creating spore suspensions of those endophytes, and this is endophyte inoculum shown here. First inoculating plants with that endophyte spore suspension. Second, inoculating plants with a pathogen like rust, moist incubating the plants and waiting for a few weeks. And then finally, the last step is to collect data on pathogen severity, and in this case, rust severity. And we can use image analysis to quantify the amount of disease found on leaves. So we're doing these types of experiments for endophytes that come from all over the study area, from these 12 different populations east and west of the Cascades. And I don't have time today to go into all of the results of those experiments, but I wanted to give you a sense of the types of questions that we're asking and the way we're answering those questions. Okay, and then lastly, I think once we have a better idea of what the fungal endophyte communities look like across this region, where there are both natural populations of Populus trichocarpa and biofuel plantations throughout, and we have a better sense of how the interactions with endophytes and pathogens differ among these dry and wet sites in the Pacific Northwest, we can investigate the potential to select particular endophytes to increase disease resistance in plantations. And I'm doing that by conducting interdisciplinary research on endophytes for reducing disease in poplar feedstock production. And specifically, I'm evaluating several different criteria that must be met before we can think about using endophytes in biofuel plantations. The first criterion, I think, is biological feasibility. So we know now that there are different endophytes that appear to reduce disease in a greenhouse setting. And then the next question is, can we also see these types of interactions in a plantation where biotic and abiotic variables are where there are biotic and abiotic factors that are more variable? So to address that question, we're working in the AFRI field trials, where we're conducting small-scale experiments to test different endophytes in the field. So plants are inoculated with endophytes that were shown to reduce rust in the greenhouse. We inoculate plants in the field. Here's our trial in Jefferson. And then we monitor natural rust infection. So there's no actual rust inoculation. We monitor how the rust naturally infects these plants that have a different background of different endophytes. And we see that in the field, too, endophytes can reduce rust, although more modestly than we saw in the greenhouse. We are also working in Pilchuck, and here we are specifically looking at endophytes that come from the Skagit site right here, which is the most nearby natural population to the Pilchuck trial. So here we're testing whether the particular endophytes from the Skagit are effective as rust antagonists in this particular trial. Okay, in addition to biological feasibility, there are several other criteria that are important for thinking about A second is environmental impacts. Now, let's say we find an endophyte, a fungal leaf endophyte that is very effective against rust pathogens, both in the greenhouse and in plantations. We want to make sure before we deploy an endophyte like that in biofuel plantations, we want to make sure that it doesn't have any detrimental effects on non-target hosts. For example, corn or wheat or alfalfa that might be growing in the adjacent field next to the poplar trees. So we're conducting a series of experiments now to evaluate how endophytes of poplar influence other hosts that poplar might interact with, both in the wild and in a plantation setting. A third criterion is social acceptability. We want to make sure that not only are the stakeholders accepting of these new technologies, these new endophyte technologies, but also that the general public has a high level of acceptability of endophyte technologies. And so to evaluate social acceptability, we're working with social scientists that are part of the Advanced Hardwood Biofuels team. And then finally, economic viability is an important criterion. Certainly, if we do find endophytes that are very effective, there will be some cost associated with their application. And this cost needs to be compared in a cost-benefit analysis to the benefits in terms of the reduction in disease. And so these types of cost-benefit analyses, these modeling tests are being done in collaboration with Greenwood Resources. And with that, I think I will stop so there's plenty of time for questions. So thank you for the opportunity to talk today, and we will field questions now.
Okay, so as you're typing in your questions, I'm going to change the screen over so you can answer some more whole questions today. Want to make everybody aware that we are having another webinar coming up on April 16th from University of California, Davis. It will be on modeling short rotation hybrid poplar plant growth and yield in the Pacific Northwest. It will also be at 10 o'clock a.m. Pacific time, so please come into that. I see people are answering the questions in the poll on your understanding of endophytes in hybrid poplar, and then whether you're likely to be discussing this with family, friends, coworkers. And so we have a couple questions here. Right now I typed in one for Posey, and then we also have one here from Gary Kaufman.
Okay, I can answer that first question about in the double inoculation experiments about waiting for the endophyte for the spores to infect the leaves. This is actually a really good question. And it gets at this question of mechanism. How in fact are these endophytes altering the outcome of disease in their plants? Are they directly interacting with pathogens either on the leaf surface or in intercellular spaces of the leaf? Or are endophytes upregulating a plant's defense genes against pathogens? The truth is here, I think there are multiple ways in which endophytes are. I think the answer is probably both. I think different endophytes are probably some might be interacting directly with pathogens. Others may be upregulating defense genes. This is an area where we're really working hard to try to understand the mechanism. But I can say for some of the endophytes that are known mycoparasites of rust, they actually parasitize rust spores. So in that case, for example, you wouldn't need to necessarily wait for the endophyte spore to infect the leaf. Because I think the interactions are actually taking place on the leaf surface. Though these endophytes, these fungi that are mycoparasites of rust, they actually were isolated from within the plant tissue. So in the wild, I think these could be endophytes that are growing inside of the leaves. When spores reach the leaf surface, those endophytes will be in a position where they can either parasitize the spores on the outside of the leaf or as they infect the leaf.
Question, which of you would you like to impact of endophytes on livestock?
That's a great question about the impact of endophytes on livestock. We're not actively working on that right now, although I do think it's something that we should consider and that we will consider as part of the non-target host effects of these fungal leaf endophytes. The group of endophytes, I mean, there are certainly many endophytes that do produce secondary compounds that are toxic to livestock animals. We are not working with that particular group of endophytes, so that wouldn't be an issue in the case of the endophytes that we're working with. But regardless, yeah, I think that we need to be thinking about how these fungi, if these fungi do infect not only poplars but other non-target hosts, what are the sort of the consequences at different trophic levels, not even in addition to livestock, to arthropods or other interacting organisms.
So for that next question about how we inoculate the poplar with the endophytes, we grow up the endophyte cultures and then we dilute it down to an optical density of just 0.1. And then we just soak the hardwood cuttings or else rooted cuttings for about overnight or 24 hours. And so it's a pretty simple inoculation.
Do we have any additional questions from our participants today? If you have questions, if you could type them into the chat box. I see that Carrie was typing. Thank you to those who answered our poll questions. How do you sign up for the next webinar? Okay, we will be sending out a link for registration soon. You can also sign up on our website, hardwoodbiofuels.org. If you go to the events page there's a link where you can sign up for the webinar on April 16th with University of California, Davis. Any additional questions? More coming in here, so we'll hang on until 11 or so, as long as people are typing. Okay, Nora's giving the link to the events page. Thank you, Nora. Thank you to Sharon and Posey for giving us fabulous information on endophytes. Very interesting. These microbes that can do fabulous things. Nora's also letting you know you can join our mailing list, and that'll give you information about all of the webinars that we will be having. After the one on April 16th, we will be having one in early May that will focus on education. And here's another question.
Yeah, this is a really good question about other pathogens like Septoria and Marsanina, and we are in fact looking at those pathogens. So far, we've or I would say till now, most of our work has focused on rust, which is the the major pathogen here in the Pacific Northwest. But starting this summer, in a series of experiments, we're going to be looking at the differential effects of endophytes on biotrophic pathogens like rust, and then necrotrophic pathogens like Septoria and Marsanina. And I think it's very possible that we might see really interesting differences between those two types of pathogens, given their contrasting strategies for acquiring plant nutrition. So rust in poplar plantations in the Pacific Northwest, I would say, was really most severe it was very severe in the 90s when this hybrid Melamsora species emerged, Melamsora columbiana, and really there was a lot of novel virulence that was generated via this hybridization between the eastern rust Melamsora species and the western rust Melamsora species. And as a result of that very damaging pathogen, or these hybrid pathogens, poplar growers in the Pacific Northwest have actually tended to shift their poplar plants to hybrids that have complete resistance to all of these rust species. And those particular plants are typically exotic hybrids. And so I think that, rust is not necessarily a huge problem now in many plantations, simply because the method to avoid it has been to go to these exotic hybrids that have complete resistance. But I think that these rust species are very tricky. They will eventually hybridize and find a way to infect the plant. So we need to be thinking about a long-term sustainable solution to diseases like rust and also like septoria and Marsanina. And I think a sustainable solution involves thinking about how endophytes have co-evolved over millions of years with pathogens to protect plants. And if we can better understand that, I think we can integrate those endophytes into our disease management strategies. Okay, so in the winter, the rust has two different hosts, and a secondary host is typically Doug fir. I mean, there are different control measures for rust. I think the number one way that poplar growers control rust is simply by planting plant material that is a non-host to rust. So again, exotic poplars. If rust is particularly bad, you can use fungicides. But again, those are not necessarily the most environmental friendly approaches to controlling an outbreak of rust. I think that's one of the reasons why using naturally occurring microbes of poplar is such an attractive option. Yes, I think that's you raise a good point that the pathogen will be able to evolve faster than trees. But that's exactly why endophytes are so great, because they actually evolve at the same rate of pathogens. So if we can find endophytes that are effective, we'll be on the same time scale as pathogens. Are there other questions about rusts and endophytes? The rust have races. So there are definitely different species of rust, and there is a hybrid rust. And I would say it's likely that we also will see that there are different races within those species. So far, we actually haven't been. We're just beginning to work on this question of looking at different races within Melamsura columbiana. So I don't have great answers right now. Yeah, so far I don't have a good answer on the races, but we are working with an eastern rust race and a western. And so when we're testing those different endophytes that come from these different populations against both the eastern and the western rust. So hopefully within, maybe 6 months or so, I'll have an update where we'll know a little bit more about the extent to which both endophytes and rust races are locally adapted to each other or to their environment or to trees. In terms of, do the fungal pathogens swamp the wild native endophytes? It's a tough question to answer, specifically because we can go out and we can sample endophytes and pathogens in the wild, but we don't necessarily. It's a little bit harder to have a mechanistic understanding of sort of what came first, the chicken or the egg question, really. So we know that both in the wild we do see leaves that are infected both with pathogens and with endophytes, and we actually do see associations in the wild where particular endophytes are associated with lower rust severity. So for example, if a leaf from the wild has a particular fungal endophyte, let's say a cladosporium species, we tend to find lower rust severity in those leaves. So that's an association, but we can't say for sure that particular isolate is responsible for the lower pathogen severity. But that's a great question. So we're looking both at mechanistic experiments, but we also want to look in the wild to see what patterns emerge. All right, thanks for all of your questions.
Okay, it's 11 o'clock here. Thank you everyone for attending. Seeing we got a couple last minute questions here. I know some of you have other things to do today, other appointments, etc. So thank you for attending this webinar. We hope to have your participation in a future Hardwood Biofuels webinar.